except for having U in mRNA instead of T. Yeah, that's correct. Here, we can accept that one. Except for having U, yes, instead of T. That's correct. The base sequence of DNA template and its mRNA molecules, no, it's not similar. TT's C TTGAT, and here AACC, that base sequence is different. As a complementary bases are there, right? So that's wrong then. An mRNA molecule of prokaryotes cannot code a polypeptide chain in eukaryote. Okay, I think we haven't specifically discussed that one, but we're going to discuss in detail in recombinant genetic technology. In recombinant genetic technology, we're going to learn the human. This is a human cell, and human cell has a gene code for insulin. It's a human cell. Human cell has a gene code for insulin. So we take this gene, separated the gene, and this gene insert into the bacteria. So this is a bacteria. And this is the human gene, insulin gene in bacteria. And this represent, the blue color represent DNA of bacteria. Now what this bacteria is doing now, this human insulin gene transcribed into messenger RNA. And the messenger RNA translate to produce insulin. So insulin is purified for the treatment of diabetes. Now we're going to learn this process in recombinant gene technology, so you can understand mRNA molecule of prokaryotes. This is the mRNA molecule of prokaryote, the red color one. Here. This is the mRNA of prokaryotes. So it can synthesize polypeptide chain of human eukaryotes. So this is wrong. Can code, right? There are 64 codons, and out of that 3 are stop codons. Then 61 left codes for amino acid. This also we learn in the genetic codes. It's a 61. It's 61 codes for the amino acid. The first triplet of base in tRNA, the first triplet of base in tRNA is not AUG. So if it is depend on the which end, 5' or 3', so if it is a 3' or 5', the code might be different. But only thing we learned that tRNA molecule. So it's ACC is the codes given, and these codes represent a binding site for the amino acid. So there is no AUG. AUG is a code for the start code for the mRNA, right? So this is then correct, third one. The next question. I think this is also not difficult question. I hope you can find the answer.
Second answer.
Yeah, non-coding sequence of the gene is called introns. And between gene, that's a gene, this is a one gene, this is a gene. The sequence between the genes, intergenic DNA. This is intergenic DNA, right? So second answer is the right one. So heterochromatins are the dense chromatin in the nucleus because they are tightly packed. Euchromatins are loosely packed DNA in the nucleus. In electron micrograph, we can see clearly heterochromatin and euchromatins.
Second answer.
Translation of eukaryotes differ from that of prokaryotes. Translation of eukaryotes differ from the prokaryotes because, right?
Yes.
It's not transcription, translation, right? Translation of eukaryotes. I told you genetic code is universal. As a result, AUG is a start codon for both eukaryotes and prokaryotes. UA, UGA, UAG are the three stop codon for both prokaryotes and eukaryotes because genetic code is universal. Today I mentioned both prokaryotes and eukaryotes, many ribosomes attached to the mRNA molecule called polysomes. Now, 1 and 2, translation in eukaryotes not occur in nucleus, right? Translation occur in the it's occur in the cytoplasm, right? So when you say nucleus, it's occur in the cytoplasm, not nucleus.
Yes.
So this is a little bit of inverse sentence. Does not start before transcription is terminated. Now we learn prokaryotes, transcription, translation happen in at the same time. Here, transcription, translation happen at the same time. But in eukaryotes, first translation happen, after transcription happen, after finishing translation happen. So does not start before transcription is terminated. Translation not start until transcription is completed. Transcription should be completed, then translation should happen. But in prokaryotes, transcription and translation happen at the same time. Okay. This is another question.
Third answer.
Yeah, we can go straight forward for this kind of a question. Variants of genes, other version, variants of genes, alternative version of genes are called alleles. So alleles we have learned alleles arise, new alleles arise due to mutations. So mutation can make new alleles, and mutation happen mostly in the DNA replication. So DNA replication mutation happens. Mutations are important for the evolution. I think again we discussed many times. Mutations are beneficial. If mutation goes wrong, it ended up a genetic disorder. But taking the risk and benefit, mutations are important for the evolutions. So we have to consider variants of genes which are called alleles arise due to mutation that occur as a result of mistake in DNA replication. Yes, that's right. This one,
Second answer.
this question seems to be a little bit of hard, as everything is same. Like we can feel that all statements are more or less correct. Now, meiosis is necessary for sexual reproduction. So, sexual reproduction, meiosis is important. Yes, that's correct. Because mitosis is important for asexual reproduction, meiosis, sexual reproduction, correct. Meiosis contribute to maintain constant number of chromosomes. That's also correct. Because it make haploid gametes join together make the zygote. That's also correct. Meiosis alternate with mitosis from generation to generation. This could be a little bit of a broad sentence. Meiosis, in that case, meiosis happen that make a haploid number n join together to n, and then mitosis make the organism make the gametes, meiosis, and again join together, mitosis. That's also correct then. So here it's a meiosis happen, and then after that mitosis. In a broad sense, there is no much problem with this kind of a sentence. Due to meiosis, same genes are transmitted from generation to generation. Now that case is not the same because meiosis, that's wrong. Because we know that during meiosis, gametes form, and the gametes have different genes, and these genes randomly are fused to make the zygote. As a result, we can see kind of different genes. But if you take other way, the gene flow, that means reproduction from generation to generation, the same set of gene existing. In that case, the sentence is true. But when it comes to variation, it's not correct because it also brings the variation. That's also half and half, like we can say correct, and this correct, this correct, this correct. Now next one, genetic recombination are possible due to meiosis. Now meiosis, prophase 1 crossing over, chiasma formation cause genetic recombination. That happen in meiosis. It's also correct. Right, now how we get the correct answer? We are not going to select the correct statement about meiosis. So we should consider, in this case, Evolutionary advantage. Now we have to focus on this word. Evolution. What is the driven factor for evolution? Driven factor for evolution is mutation. Mutation is number one. Then what are the other factors? Bring variation. Now mutation is the number one factor which bring the variations. But mutation is not only factor bring variations. Variation can bring random assortment. Random assortment happen in the metaphase 1. Recombination or crossing over happen in prophase 1. And random mating. Now these are the other factors which bring variation. So mutation is important for the evolution. That's the number one factor. Other factors are random assortment, recombination or crossing over, and random matings. Now, random recombination, this happen in meiosis. So then number 5 is important for the evolution. So variations are important for evolution, then number 5. So this question is quite good question. It really address certain, it's very focused, it's very specific. So we have to carefully think about the evolution and the relationship of evolution and meiosis. So meiosis and evolution, the link is metaphase 1, random assortment of the chromosome, prophase 1, crossing over, and recombination. Keep in mind, in biology, as you don't see in a local GCE syllabus very much highlighting that, but in most of the biology, all the books, these two are very important events for variations, to bring the variation. So these bring the variations, and mutation always appear as the first place to make the variations and new alleles. Is this clear?
So if meiosis does not maintain a constant number of chromosomes, from generation to generation, when the chromosomes increase,
Second one.
no. Yes, some species.
This actually second one, if you take animals, not the plants, the gametes, let's say human. So human has 46 chromosomes. So each generation, human has 46 chromosomes. So meiosis make 23 chromosome gamete, 23 chromosome gamete joined together to make 46. So human chromosome number is 46, which is constant due to meiosis, because meiosis make gametes with 23 chromosomes. Imagine that meiosis doesn't make this number, it make like a 24 and 26 like this, then our chromosome number change. So that's why second sentence is correct, but it has nothing to do with evolution. The thing is, this sentence has no contribution for evolution. It's correct, but no contribution for evolution. And this is correct, but no contribution for evolution. So we have to find correct sentence linked to the evolution.
So in the resource book, they have said that maintain a constant number of chromosomes from generation to generation maintains genetic stability. Stability, saying that because the number of chromosomes increases then from generation to generation.
Yes, stability is this one, keeping a 46 chromosome in human. So it doesn't
So
change the chromosome number.
if that number increases, won't it be a disadvantage for evolution?
The number change, if this number change, uh, this can cause various problems. One is it could lead infertility because odd number of chromosomes, organism with odd number of chromosomes cannot reproduce. For example, if there's an organism with 47 chromosomes, they are infertile because they cannot make a even number of chromosomes. So that's not evolutionary advantage, but we can see this in plants called polyploidy. We discussed this in previous section, polyploidy. So that certain plants, their chromosome number increase, like in wheat, strawberry, and some other plants. They have a tetraploidy, octaploidy like this. The chromosome number increase. So increased chromosome number is not evolutionary advantage because lower animals, lower than human, they have a more chromosomes. So having more chromosome is not the evolutionary advantage because if you look certain animals, even I think sheep and goat like a 64 chromosomes, like they have 64 chromosomes. Their chromosome numbers are even some animals they have a high like 100 chromosomes. Uh, the increasing or having more number of chromosomes doesn't make evolutionary advantage. The advantage comes with new alleles. So previous question, this one. So alleles, variations of genes which are called alleles. So alleles are the advantage of evolution, not the chromosome number. Many alleles, more alleles than more chromosome is advantage evolution.
So say if meiosis doesn't maintains the constant, like 20, it will go from 46 to 92. That's disadvantageous. So even second one can be right, no? Then
Meiosis make 90, 90, 92?
no, if it doesn't maintain constant number, when the zygote and the egg get together, always it will double, right? The number. That's a disadvantage, right? Then for the next generations, evolutionary.
Yes, yes. Having a more chromosomes. Uh, if you take only human, like an example, human, this doesn't make a perfect human. Like we have to talk about next section, the mutation. Let's say a person has 47 chromosomes. Is it? This is possible. We have people with 47 chromosomes. There's one additional chromosomes. It's not 46, 47. But these people ended up with genetic disorders. So it cannot stabilize the genetically, having more chromosomes. Like Down syndrome, they have a 47 chromosomes. Additional chromosomes are there. So they have a problem, like it's not advantage evolution. So stable chromosome number is advantage. Having more chromosomes or less chromosome, disadvantage. So we can easily think having more chromosome is advantage because more alleles, it brings more alleles. But somehow this make a big genetic stability. It cannot probably continue the generations. So most of the time the inviable or infertile or unstable. I think the clear approach will come evolution advantage. Evolution. When you say evolution, evolution is possible because of variation. Evolution happened because of variation. Variation happened because of mutation. Variation happened because of random assortment and recombination. So then we can find direct link evolution, variation, and genetic recombination. If you thoroughly understand this concept, it is just like this question. You see clearly the third one is correct answer despite of other factors. So you can analyze other things here and there, and you have you can take time. But when you see third one, crystal clear, this is a correct answer. So if we understand evolution and mutation concept, then meiosis contribute for variation only prophase 1 and metaphase 1, random assortment and crossing over. These two events are the main step bring the variation. And the variation is driven factor in the evolution. So that likewise you can directly link question number answer number 5 with the questions.
Yes.
Yeah, I think that's it. Some of the question I found. Yeah, at least 1, 2, 3, 4, 5 questions. Okay, so we want to finish this section about protein synthesis. We're going to discuss briefly the fate of the protein. I would write the fate of the polypeptide then. So polypeptide is produced at the end of a translation process, and we're going to see what happened to this polypeptide. Briefly, we all know that polypeptide ended up in the functional proteins or structural proteins. So it become at the end protein molecules which serve as any enzyme or any function, or maybe it could end up in structural proteins such as collagen-like proteins. So we're going to see that process, how the value added to the polypeptide chains, how the value added to the polypeptide chain, and how this polypeptide chain further process in the cell. Okay. So how we can see this polypeptide chain for the learning purpose. At the last part of the translation, we have seen large subunit of ribosome, a small subunit of ribosome, and then last tRNA molecule holding the polypeptide chain, and then the messenger RNA. There are several components we have seen at the end of the translation process. For example, this is the large subunit, this is the small subunit, and this is the mRNA molecule, And this is the tRNA molecule still holding the polypeptide chain. And this is the polypeptide chain. Now, coming back again, where we can find this ribosome, we know that the most of the ribosomes attached to the rough endoplasmic reticulum. There are free ribosomes and also ribosomes attached to the endoplasmic reticulum. For example, so this is the endoplasmic reticulum. So I'm representing part of the endoplasmic reticulum. Okay. Now this polypeptide chain, it has a region. This region, for example, this region, it serves as a signal peptide. So this part is a signal peptide. Now, the polypeptide, this polypeptide chain is not directly one-to-one going to make the protein. It just going to change the polypeptide chain at different levels to get the final version of protein. So it has a different region, like these regions are not necessary sometimes for the protein, final protein, but they are there to make the process to the end. Now the signal peptide guiding this polypeptide chain to the endoplasmic reticulum. So this is the endoplasmic reticulum. Okay, next. What's going to happen? This signal peptide can bind to a special protein molecule called signal recognizing particle, abbreviated by SRP. SRP. Now I just briefly, we don't want too much detail. So next step, I will explain you very briefly. And once it bind with the signal recognition particle with the signal peptide, this polypeptide is guiding towards the endoplasmic reticulum. That means endoplasmic reticulum has a receptor or transporter, and to the transporter, signal recognized particle bind, and this is still attached to the signal peptide. And briefly, the rest of the polypeptide is moving to the endoplasmic reticulum. So it's moving to the endoplasmic reticulum. After that, it become more independent in the endoplasmic reticulum. Okay, can you, if you remember, can you give a few functions of endoplasmic reticulum?
Transportation of substances.
Okay,
RNA.
transporting of substances.
Yeah. Ariana asking.
Rough endoplasmic reticulum, yes.
Then synthesized proteins like transport the synthesized proteins are the ribosomes.
Yes.
And provided secretions to the Golgi complex to the face of it.
For physical transport, okay. So how many you listed? 3, right? Transport of protein, right? Transport of protein.
Yes.
Yes. And also transport of vesicles. And another one you mentioned, right? Synthesis of glycoproteins, right? The other one,
Yes.
glycoprotein. Now, in that case, transport of protein. Actually, ARIA, if you look the resource book, it doesn't mention the main, one of the main function of ER. So it listed the function of ER, but unfortunately, it doesn't mention one of the main function of ER is protein folding. Now, one of the main function of ER is protein folding. Now, if you look primary, secondary, tertiary structure of proteins, you can understand how important this process to the protein molecule. Now, check. If you recall, primary structure is the sequence of amino acids. Secondary structure is the beta pleated sheets and alpha helix made by the hydrogen bonds. And the tertiary structure is a functional 3D shapes, globular shape of the protein. So it needs folding with making ionic disulfide, hydrogen and hydrophobic van der Waals bonds. They all bonds important to make the final version of the protein. Now the protein folding take place in the endoplasmic reticulum. That means these proteins, like here, they start to fold. Now the folding is not a spontaneous process. It's done by many molecules, many systems involved in the ER to get the right shape. So we learn this has a disulfide bond, this has hydrophobic bonds, this has a hydrogen bonds, and this has ionic bonds. So many bonds form between R groups, and finally it gets the right shape of the proteins. So this protein folding takes place in the ER.